Thanks for jumping on a call today, Rob. So we have Professor Rob Malenka here, who is the Pritzker Professor of Psychiatry and Behavioral Science at Stanford. And yeah, we're talking today about a really interesting preprint, a preprint that Rob and a number of co-authors have published under the acronym Psy-BAANC, for the Psychedelic Bay Area Animal Neuroscience Consortium. Summary, this is a preprint looking at the replicability of psilocybin in a number of different mouse behavioral models. And it's used a single dose of psilocybin, 2 milligrams per kilo IP, which is commonly used. The same strain of mice, black 6 mice of the same age, both sexes, which is nice because that's not always done in neuroscience. And I thought what was really neat is that besides using a very large number of mice for replicability, it is conducted by 5 different labs around the Bay Area, right? Featuring sort of key behavioral labs from UCSF, UC Berkeley, and Stanford.
Yeah, I think you summarized it well. The goal was basically based on my very long academic career. I've been an academic neuroscientist, yikes, for almost 45 years. And, I'm sure any of your listeners who are academic scientists or scientists within biotech or pharma do appreciate that one challenge in discovery science is the replicability and reliability of findings. I know historically in my career, I have tried to replicate prominent findings in various fields and have had great difficulty, which is often very frustrating. And because you can't build knowledge and you can't build on exciting findings unless they are, in quotes, replicable. And that term can mean different things to different people. What I mean by that is that independent labs, not the lab that first reported a finding, can, to their best of ability, follow the methods in whatever experimental approach and assay is being reported, and basically get the same result, at least qualitatively, if not quantitatively. And unfortunately, it turns out that a lot of findings don't meet that criteria. And in particular, as many people know, some of the most exciting findings published in the most prominent journals often don't meet that criteria of replicability. And that does not mean that the original investigators were fraudulent or were incompetent. I think it's largely a reflection of biological noise. The more complicated the assays one is doing, the harder replicability often is. Then it also has to do with the sociology of academic science now, the incentive structures in academic research, which often is quite different than the incentive structures for research in biotech or pharma. But in academia, you want to be new, innovative, and creative. And replicability, in my view, and I'll be a little provocative here, I think the journals and funding agencies, at least in the United States, I do think they care a lot about it, but I think the way they try to implement rules and regulations to maximize replicability show a very clear lack of understanding of what the real practice of science in real academic labs entails. And the way they try to incentivize, demand, and implement replicability is failing. And so I was given the opportunity of being able to raise some money to put together a consortium of labs. They happen to be in the Bay Area because Stanford is in the Bay Area. And even though I could have set this up and included labs from around the country and even around the world, I thought as a first attempt, there were a lot of advantages to having the labs in the same relative geographic location so we could meet in person when needed to meet, even though we all know we can Zoom or... And so I got a group of investigators, truth be told, all of whom I knew, 2 from Berkeley, 2 from UCSF, me and Boris Heifetz from Stanford. And we talked about what studies could we do, beginning with trying to understand what are the replicable effects of
Hmm.
psilocybin in very simple behavioral
Hmm.
assays in mice. You,
Mm.
which we thought was the simplest and most straightforward topic to begin to address.
Hmm.
And that generated the paper, which is on bioRxiv, behavioral effects of psilocybin that outlast what we call the acute experience, that after the substance is presumably no longer in the brain, what effects could we see a day later or a week later?
Yeah,
And
and this is an important aspect of psilocybin's effects in the clinic,
of
right? Is they're
course,
rapid
that's what
and
everybody
post-acute.
interested in for potential therapeutic benefit. And we didn't see a lot of long-term
Mm.
effects. And so we actually expected this. All of the experiments were done in so-called control mice, meaning mice that did not have any obvious deficits in the behavioral
Mm.
or pathological changes in the assays we were doing. And since a lot of the previous work in psilocybin had been done in models of depression or mice that had been stressed in this way or that, we have now been asked to go back and at least do some experiments in mice that have been stressed by some protocol, which we are
Right.
going to do and we are happy to do.
Very
All
cool.
right. So, and again, I mean, our goal for this effort was several fold. We wanted to use it as another example of how cooperative, collegial, collaborative science can produce replicable results as a model, not only for the psychedelic science field, but other fields. And the pressure to produce replicable results is not there yet. And
Mm.
that I want to make it clear that isn't only because it's a psychedelic science research topic. That will apply to any new research endeavor, many of which I have been involved in. But because of the attention psychedelic science research is getting, and because of its importance as potential therapeutic drugs, I and the team of investigators I brought together think it's more important because
Mm.
people are generating hypotheses that they are claiming apply to human beings who take these substances, often based on very limited data,
Hmm.
based on single papers. You just learn the hard way that
Mm.
you don't want to put too much emphasis on single publications and the
Yeah.
conclusion of single publications until the key conclusions, the key results have been replicated by several different labs.
Mm.
And I would like to believe I have a decent reputation in the scientific world because a lot of my most prominent results from my lab have been replicated by many other labs and have provided foundations upon which other labs can build.
Based on my understanding of the science at hand, they seem to make some pretty grandiose claims that have not yet been replicated. And I stand by that as well, that, extraordinary claims require sort of extraordinary evidence. That's a pretty good position to take in the sciences, which is exactly why I was such a fan of this preprint. I think anyone who's studied psilocybin preclinically in mice or rats even, psilocybin has received a lot of attention, as you mentioned, because it's extremely interesting as a tool. It's also extremely interesting in psychiatry as a potential therapeutic. And there's a huge amount of sort of discord in the preclinical literature, especially about exactly what it's doing in mice. So with that in mind, given this is one of the, I think it's fair to say this is the largest study of mouse behavior with psilocybin. Were there any things that were particularly replicable from the preprint that you recall?
And again, I want to make it clear, all of these behaviors were done in quotes, normal mice, not mice
Mm-hmm.
that had been stressed. So not in models of depression or,
Mm-hmm.
or other neuropsychiatric disorder you want to mention. And we did that. We had extensive discussions where we should begin, and we just decided it would be useful on basic principles to start with, in quotes, normal mice brought up in a very restricted environment in a typical animal research housing facility in an academic institution where mice are housed, whatever the number is, 6 or 8 to a cage. And, the most reliable effect was the acute effect in so-called anxiety measures.
Mm-hmm.
And the acute experience of the psychedelic was anxiogenic, that is, increasing anxiety during the acute experience, meaning animals were administered psilocybin IP at 2 milligrams per kilogram, and then 10 to 15 minutes later, they were placed in an open field, they were placed in an elevated plus maze, and they spent less time in the center of an open field. They spent less time in the open arms of the elevated plus maze, which in our simplistic anthropomorphic way, we think as indicating the animals were more anxious, more nervous about the environment in which we're experiencing. I should also say the head twitch assay was highly reliable between all 5 labs.
To be clear, the EPM is a good example where certainly other labs, a lot of discord in that particular model, because other labs have certainly shown more time in open arms, so-called anxiolytic behaviors.
I'm actually looking at the results, and what I like about our results is despite the fact that there was some variance in the baseline measures in terms of, how much time this lab found the animal spent in the center of an open field, or how much time they spent in an open arm and elevated plus maze. The direction of change during the psilocybin experience was pretty consistent.
Right.
And to me, and I'm just looking, when I say consistent, it means either all 5 labs got the same directional result, or 4 out of the 5 labs got the result. So that was probably, and that in the head twitch was everybody saw increases in it with actually very similar time courses. And then, in the novel object recognition during the acute phase, we saw a decrease, presumably because the mice were tripping to some extent. What was disappointing is we basically did not see any consistent or significant, what we call persistent effects. That is, following 24 hours after giving the psilocybin, we did not see any changes in these motivational assays, the tail suspension test and forced swim test. We didn't see any effects on sociability doing the 3-chamber social preference assay, which was disappointing. We wanted to see an effect. We didn't see any consistent effect on the extinction of fear memories. And there have been several papers reporting that. Between labs, that was probably the most variable result. There was one lab that really did seem to see an enhancement of fear extinction, but 4 other labs did not. And, we present the data as one would expect, honestly. And the point is, any lab can get a result that looks real and not do anything wrong, because behavioral assays are enormously inherently variable.
Can I ask, I think I saw Grant Glatfelter and Mike Bauman had a recent paper where they showed, it's amazing how many people don't look at sex differences in the neurosciences, and this receives a lot of criticism, but did you find a difference in something more replicable like head twitch response across sexes with such a large group?
There were some modest sex differences.
Right.
But they were modest, and to be honest, I have to look at the paper.
Yeah, that's totally fine.
There were not dramatic differences where we saw a result in one sex but not the other.
Right.
And then again, some of this is statistical. Like, I'm looking at our data, and there was the amount of head twitches in females in response to psilocybin was quantitatively slightly larger than in male mice.
Right. I think that's what others have reported with different drugs as well, other psychedelics. I think maybe DOI, there's a report that females have slightly high head.
I have to be honest, we were hoping for more effects, and effects that outlasted the acute experience, the persistent effects. We did do one assay of a very prominent paper that has reported that different classes of psychedelic drugs open a so-called critical period for social reward learning.
Yeah, this is Dolan's paper, right? I think people will be
And
familiar with
we
that one.
did a very assay that we think addresses the same topic. That is the assays the rewarding component of a social experience, but we designed a different assay that we think is much easier to perform, with the goal that other labs can look at our methodology and see whether they can replicate our findings or not. So I'm a big advocate for, let's start simple. So in terms of this social reward learning, which is still a very complicated phenomenon, we developed basically an assay built on simple drug elicited condition place preference, and a much shorter assay, and we think much simpler one to perform. And what we report is that we can get a social reward result in younger animals, consistent with the Gould-Dolan paper. We do also see that the ability to elicit a place preference in response to a social experience that is social reward disappears at older ages. At least we can't detect it. We used a higher dose of psilocybin, but I would argue it's an important assay to do. And if any of your listeners happen to be mouse behavioralists, behavioral neuroscientists who are interested in this topic, I encourage them to do it themselves.
It's a super rich preprint. I think there's a lot of really good behavioral work in there, and I'm excited to see it in press once the reviewers done with the request. What's next for Psy-BAANC, if I can ask? I'm sure that there's an endless set of possibilities here.
I'm hoping to gather the labs in the next 2 weeks to decide on the next set of experiments in response to these reviews, and it will certainly involve doing some stress manipulation, whether it's chronic mild stress, whether it's chronic restraint stress, whether it's administering corticosterone in the water or with one of these subcutaneous pellets. The group will decide. And so we will certainly do some stress protocol. We will do, the standard primitive assays, tail suspension test, sucrose preference test. We'll have to decide whether we want to do some anxiety measures like elevated plus maze, with the hope that the chronic stress protocol changes the behavior of the mice. And if we get that, then compare saline with a psilocybin treatment to see if we ameliorate these behavioral changes or not. And then once we get that done, my hope is to be able to generate more funding for this group and perhaps add in some other groups and decide what are the important assays we are interested in. And should we stick to just studying psilocybin, or should we expand into studying other agents? And should we do some mechanistic studies as a group?
That's another good point. People sort of under this catch-all of psychedelics treat these as sort of a pharmacologically homogeneous group of compounds, but we know preclinically and clinically they seem to do pretty different things depending on the specific drug as well.
I mean, obviously there's decades of work to be done, actually, on these class of substances and how they work and the behavioral changes they may cause. If we find that psilocybin doesn't cause as robust changes in the behaviors we are interested in, at least in mice, then I will be pushing the group to change the substance we're studying. It's much more fun to study something that causes a robust change.
Sure.
That's really what we want. So we'll see.
Awesome. Well, I thought it was an excellent preprint, a really great contribution to the field, and I'm sure a lot of others feel the same. So, looking forward to seeing it in press. And thanks so much for your time, Rob.